Welcome to topic 2.4 of module 2. In this particular topic, we will learn about active transport mechanisms. During the course of this video, try and answer as many questions as you can from module 2, topic 2.4. The learning objectives for this topic are to describe active transport and differentiate between primary and secondary active transport, to describe the process of active transport using the sodium-potassium ATPase pump as an example, and understand why this pump is ubiquitous in cells, and to differentiate between non-specific and receptor-mediated endocytosis, phagocytosis, and pinocytosis. Whenever a cell uses energy to move ions and molecules across the membrane, the process is referred to as active. Substances that are moved by active transport across plasma membranes are usually molecules that are unable to pass the membrane by passive transport. This can be due to reasons such as the molecule being too large to pass through the channels, or ions that cannot diffuse through the membrane. Active transport, like carrier-mediated facilitated diffusion, requires protein transporters to move substances across the membrane. In active transport, solute pumps move solutes, most importantly ions, against a concentration gradient. That is, from an area of low concentration to an area of high concentration. And for the cells to do this, they need to expend energy constantly. Active transport opposes diffusion, which is the process in which solutes move from high concentration areas to low concentration areas, without consuming any energy. Based on their source of energy, active transport processes are distinguished into two types. In primary active transport, the energy to move solutes across the membrane comes directly from the hydrolysis of ATP. The sodium-potassium pump is the most common example of an active transport system. In secondary active transport, the movement of substances is driven indirectly by the energy stored in ionic gradients created by the operation of primary active transport pumps. The sodium-glucose co-transporter in the gastrointestinal tract is one of the examples of secondary active transport. The sodium-potassium pump is one of the most extensively studied primary active transport systems. These pumps constantly operate in living cells, moving substances against their concentration gradient. The sodium-potassium pump plays a key role in several functions of our body, including maintaining the membrane potential in nerve cells and controlling the cell volume. The concentration of potassium inside the cell is roughly 10 times higher than outside the cell, and the reverse is true with regards to sodium. Because of the difference in these concentrations, sodium and potassium leak slowly but continuously through leaky channels in the plasma membrane down their concentration gradients. To counteract this inward movement of sodium and outward movement of potassium, the sodium-potassium pump operates almost continuously as it drives sodium out of the cell against its concentration gradient and pumps potassium back in. This process of continuously moving sodium out of the cell and potassium into the cell across the plasma membrane requires a constant supply of energy. As a result, these pumps expend almost 1/3 of our total caloric intake. The sodium-potassium pump not only preserves the chemical content of the cell, but also indirectly controls the volume by preventing water from entering the cell through osmosis. If the ATP levels were too low for the pumps to operate, sodium would gradually diffuse down the concentration gradient into the cell. Because of increased sodium concentration inside the cell, water would enter the cell through osmosis. Now let's learn about how the sodium-potassium pump operates. The sodium-potassium pumps are situated in the plasma membrane. It constantly pumps sodium out of the cell and potassium back into the cell. To start with, 3 sodium ions within the cytoplasm of the cell binds to the protein carrier. Hydrolysis of an ATP molecule then provides the energy to change the shape of the carrier protein. This change in shape allows the sodium to be released on the other side of the membrane, out of the cell and into the extracellular fluid. One phosphate group from the ATP remains bound with the protein. This new shape of the protein has a high affinity for potassium ions. Two potassium ions from the extracellular fluid then bind to the protein, which again causes the protein to change its shape. This conformational change allows the release of the phosphate group, which in turn allows the protein to go back to its original shape. As a result, the potassium ions are released into the cell. The protein in its original shape has a high affinity for sodium ions, and when sodium ions bind to the protein carrier again, they initiate another cycle. The most important feature of this pump is that both sodium and potassium are moving from an area of low concentration to an area of high concentration, which means that each of these ions are moving against their concentration gradients. Movement of ions through this type of transport system can only be achieved by constantly spending ATP. The secondary active transport is driven indirectly by a concentration gradient created by primary active transport. Because of the sodium-potassium pump of the primary active transport system constantly pumping sodium out of the cell, it creates a low concentration gradient. In secondary active transport, a substance such as glucose is pumped from a region of low concentration to a region of high concentration. This process requires energy because glucose molecules are transported against their concentration gradient. However, the energy that drives glucose across the membrane does not come directly from ATP. Rather, it comes from the low sodium concentration gradient created using ATP by the sodium-potassium pump. Because ATP does not fuel the carrier proteins directly, they are called secondary active transport proteins. Our gastrointestinal tract is designed essentially to absorb all the available glucose from the gut lumen. But because the concentration of glucose inside the cells lining our gastrointestinal tract is higher, glucose from the gut lumen must be moved up its concentration gradient into the cells using secondary active transport. The sodium-potassium pump constantly pushes sodium out of the cells. This creates a concentration gradient of sodium pointing towards the cell. As a result, sodium diffuses back into the cell by binding to a membrane co-transporter protein. As sodium diffuses back across the membrane, it takes glucose with it against its concentration gradient and into the cell. Sodium is then pumped out by the cell again by the sodium-potassium pump. As the glucose concentration increases inside the cell due to the secondary active transport, it diffuses out of the basal surface into the interstitium via facilitated diffusion using glucose transporters. The other major means of active membrane transport is vesicular transport, which uses membrane proteins to form vesicles and ATP to transport substances. Non-specific endocytosis, receptor-mediated endocytosis, phagocytosis, and penocytosis are all examples of vesicular transport. In non-specific endocytosis, fluids containing large particles and macromolecules are transported across the cellular membranes inside bubble-like sacs called vesicles. Vesicles move substances into the cell by endocytosis and out of the cell by exocytosis. Receptor-mediated endocytosis is the main mechanism for specific endocytosis of most macromolecules in the body. This selective mechanism allows cells to concentrate material that is present only in small amounts of the extracellular fluid. The receptors for this process are plasma membrane proteins that bind only to certain substances. Enzymes such as insulin and other hormones are taken up by the cells using receptor-mediated endocytosis. Interestingly, viruses also use the same mechanism to invade the cells of our body. In phagocytosis, the cell engulfs some relatively large, solid material, such as a clump of bacteria or cellular debris. When a particle binds to receptors on the cell surface, cytoplasmic extensions called pseudopods flow and form around the particle, eventually engulfing it. The engulfed particle is then digested in a vesicle called a phagosome. Phagocytosis is commonly seen in macrophages engulfing bacteria. The vesicular transport process that ejects substances from the cell's interior into the extracellular fluid is called exocytosis. Membrane-bound vesicles with secretory substances inside them are transported and released into the extracellular fluid using this process. Common examples of exocytosis are the release of neurotransmitters and the secretion of mucus. In summary, from this video, we have learnt about the primary transport process and how the sodium-potassium pump works. We have also learnt about secondary transport processes and how glucose is transported into the cell using the sodium-glucose transporter. And finally, we have also learnt about the other mechanisms of vesicular transport, such as endocytosis, phagocytosis, penocytosis, and exocytosis.